Okay, I want to just review the lesson Life from Life from the second week of our course, our life science course. And this lesson began with the strange questions: how do you make a mouse, and how do you make maggots? And these are strange questions to us nowadays, because we know that we can't make mice and we can't make maggots. We can maybe attract them, but we can't make them. But several hundred years ago, that wasn't obvious to people. So that was what we began thinking about. How people came to understand that life comes from other living things, not from non-living things. Now this is a strange recipe from the 17th century. It's a recipe for mice. How to make mice. You get some old sweaty underwear, and you get some wheat, and you put them together in an open container and leave them for 3 weeks, and from that mixture you will get mice. And rather similarly, here's a recipe for maggots. You get a bit of meat, you leave it in the open, and after a few days, a week or so, then you will get maggots. So that's how to make maggots. Now, we think these ideas are ridiculous. We know that that is not really true. We can't make mice from underwear, and we can't make maggots from meat. But the question I put to the students was, how could you test these ideas scientifically? But before we thought about that, I just wanted to make clear what the objectives of the day's lesson were. And the lesson is called Life from Life, and the first objective, the first aim, is that students by the end should understand the ideas of biogenesis and the idea of spontaneous generation. Second aim was that we should be able to describe the steps in the scientific method, the method that most scientists use when working. And thirdly, that we can describe a controlled experiment that disproved the idea of spontaneous generation and led to the idea of biogenesis. And we looked briefly at the scientific method, and I wanted to say that this is not the method used by all scientists all of the time. What is closer to the truth is that parts of this are used by most scientists most of the time. So the scientific method begins with some problem to be solved or question to be answered, and then usually some research, gathering information about the problem. After that, there's some idea, some prediction, some idea of what you think will happen before you go on. Then, in order to test the hypothesis, so this prediction is called a hypothesis. In order to test this, we set up an experiment. And after we carry out an experiment, the results are analysed, and the scientist is able to draw conclusions. And from the conclusions, he or she is able to say, is the hypothesis true or is it false? And if the hypothesis is false, then the next step is to come up with a new hypothesis and test it again with a new experiment. Then we looked at a famous experiment which was carried out several years ago, which used the scientific method to answer the question, do maggots come from non-living thing? In other words, the meat, or do maggots come from living things? In other words, flies. And I won't talk through this in detail, but the pictures are there for you to study. Meat in open jars, meat in sealed jars, and meat in covered jars. And you can see for yourselves what the results were here. And from the results, it was concluded that the maggots came from flies, not from the non-living meat. In response to that, some students said, well, why would we want maggots anyway? But that's not really the point. The point is that there's a big question here. The big question is, does life come from non-living stuff, or does life come from other living things? And the words to describe these different ideas are, life come from non-living stuff is called spontaneous generation, and life coming from other living things is called biogenesis. So another example, broth, basically you can think of it as just being a soup. If left open to the air, will be full of bacteria after a few days. So the question is, where do bacteria come from? And this question was answered by a French scientist, Louis Pasteur, and what I wanted students to do in the class was to look and read about his experiment, which could be found on page 20 of the textbooks. So just to repeat, what I want students to do is to write in the science notebooks definitions of spontaneous generation and biogenesis, and describe the experiment of Louis Pasteur. And we ended up by summarizing his experiment. So here's a summary of Pasteur's experiment. The key thing in his experimental design was that he used these flasks with these long S-shaped necks. He started off by boiling the broth in the flask. The broth is this brownish yellow stuff here. Started off by boiling it to kill all bacteria. One flask was left upright, and another flask was tilted so that the broth went into the neck of the flask. And another flask had its neck broken so that it was then open to the air. And the results were as follows. The flask that was left upright, the broth did not become contaminated. The flask that was tilted, the broth did become contaminated. And the flask which had its neck broken also became contaminated. And how do we understand these results? We understand these results by saying that bacteria in the air in an upright flask cannot travel down and then up and then down again to get into the broth. But if the flask is tilted, the broth can come in contact with the air and there is bacteria in the air. Similarly, a bacteria in air, if the flask's neck is broken, can fall down into the broth and then can grow in the broth here. And the interesting fact that I mentioned in class is that this experiment was done around 150 years ago, and I believe some of Louis Pasteur's original flasks with broth still in them are on display in the Museum of the Pasteur Institute in Paris, and the broth is still sterile after 150 years. So the big idea of this class was, through a series of experiments, scientists understand that life comes from life. Life comes from other living things, which is the idea of biogenesis.